Welcome, everyone, to session 8, the chemistry and the origin of life. We'll be talking about topics in chemistry and how they relate to how life began. We're not going to cover, of course, the entirety of the field of chemistry in this one session, of course, but we'll introduce some ideas that make the case for a supernatural original life or life from intelligent design. For this session are The Mystery of Life's Origin by Thaxton, Bradley, and Olson, Signature in the Cell by Dr. Stephen Meyer, or you can watch the DVD Unlocking the Mystery of Life if you'd like a video presentation of the problems and the enigmas of the mystery of life or the origin of life chemistry. Now, if you really want to take on a deep dive into chemistry and you are familiar with some basic chemistry and want a really deep dive into the problems of origins of life chemistry, and want to go into in a more complex presentation or more detailed presentation, you can look up Dr. James Tour's uh, YouTube channel and go back about 4 years, and he has a 14-part series where he covers a lot of the major problems of origin of life chemistry. It's a challenge. He speaks as a chemist, not as a layperson in those videos, but it's very revealing and educational about the tremendous problems and enigmas from getting from chemicals to life. But definitely worth doing it if you want to really do a challenging study. So let's begin at the beginning, of course, with what does the Bible say about the origin of life? Well, let's look at Genesis 1. Then God said, "Let us make man in our image, after our likeness, and let them have dominion over the fish of the sea, and over the birds of the heavens, and over the livestock, and over all the earth, and over every creeping thing that creeps on the earth." So God created man in his own image. In the image of man, he created him. Male and female, he created them, and God blessed them. So, is in his image is not said about any other living creature. No. No other biological entity is made in God's image, only humanity. So, what does this mean for us and our value before our Creator? As stated in previous sessions, origins matter and matter a lot. Our origins determine much about who we are and our value. Let's look at Genesis 2:7. Then the Lord God formed the man of the dust from the ground and breathed into his nostrils the breath of life, and the man became a living creature. The key takeaways there: God created life. Life did not spawn from the ooze in a warm little pond. God created man also fully formed. There is nothing from Scripture that alludes to gradual change from a simpler form. And that would be called theistic evolution, God creating a simple form of life and then allowing evolution to produce a human being later. Scripture doesn't say that. It doesn't allude to that, and there's no evidence within the Bible for a theistic evolutionary point of view. So the answer of the origin of life lies in chemistry. What we will do is examine a few of the enigmas of origin of life chemistry and see where the evidence leads us. So I'm going to present these as enigmas or puzzles. Well, let's talk about the first one. There's a problem that I will call oxygen in the atmosphere enigma. The scientific journals will support that the Earth has always had a lot of oxygen in the atmosphere. Well, so what? Well, the problem is oxygen is actually highly corrosive. And that's why we need to consume antioxidants in our diets to help our body repair from what's called oxidative stress or uh, oxidation of the molecules within our cells. So if oxygen is a corrosive, which it is, and it was present as the early building blocks of organic molecules were being formed, then those organic molecules would not last very long and they would fall apart very quickly because they would oxidize. The oxidize would corrode them and those molecules would fall apart. So we have the energy enigma. Now, the energy enigma is one of many. And what do I mean by that? If you did the math and take a look at just purely the mathematics of the energy of the cell, how much energy is expended, how much energy is consumed, how much energy is locked away in chemicals, our cells should absolutely explode, but they don't. There's also a problem with energy is in order to be used in the cell, it has to be just the right kind of energy. Not too much, not too little, the right type of current, uh, etc. So the control of that energy from the origin of the very first life is an enigma. How did that control over the energy of the cell evolve? Because if it wasn't there at the beginning, then our cells should just explode or catch on fire. There's so much energy within them. So now the information enigma. What do I mean by that? I'll explain that later. That is our key concept for today. But DNA has was discovered, and then the function of DNA was discovered that it is an information storage medium. Well, think of it as the hard drive of the body. So the enigma there is, where did that information come from? How does nature assemble long strings of very complex, specified and functional information? The chirality enigma. Well, what's that? The chirality in chemistry means handedness. What does that mean? There are some molecules that are left-handed, there are some molecules that are right-handed. And the body will only use purely left-handed or purely right-handed, depending on the kinds of molecules the body wants to build or the cells want to build to keep the cell alive. Well, if you have a bunch of molecules that are the wrong handedness, uh, if you're trying to build with left-handed molecules and there are those right-handed molecules floating around the solution, those right-handed molecules interfere with building of the molecules that the cell needs. So how does nature purify the mixture? How does nature remove all the wrong-handedness molecules so you can go to the next stage and build the next larger organic molecule? So the biopolymer enigma, well, that enigma is uh, comes in when you start having the building blocks of life, amino acids and so forth, ribose for certain molecules, as an example. But then you have to start connecting these building blocks into extremely long chains. Extremely long chains. Some of those molecules are humongous. They can be 3,000 amino acids long. Well, how do you keep that long biopolymer together, and how do you connect all the strings or all the individual building blocks without the molecule falling apart as you're building it? So the putting together of the building blocks into these huge long molecules and keeping them protected is an enigma if you don't have a living cell all ready to do that. Where did that evolve from? So the water enigma. Well, that sounds strange, and it is a bit strange that we need water to stay alive. Biological life requires water, yet water is a problem for the formation of the first life. Why? Because of a um, law of chemistry called hydrolysis. If you place organic molecules into water, water is a universal solvent. Water will begin to attack the bonds of those molecules, split them apart. So water, although we need it for life, is an actually very, very poor medium by which to form these delicate organic molecules and keep them together over long periods so they can quote unquote evolve. So water is actually an enemy to life as well. So the protein synthesis and enzyme enigma, that's a mouthful, sounds strange, but in order to build a protein, you have to have enzymes to help build that protein. Well, you can't build the protein. But what are enzymes? Enzymes are a type of protein molecule. So in order to get an enzyme, you have to have proteins first. But you can't have proteins first unless you have an enzyme to build the protein. So you have a chicken and egg problem where the enzymes and the proteins have to quote unquote evolve or have to be built together at the same time, then work together. One can't evolve and then the other evolve. They have to be uh, together at the beginning and functioning. So the protein fold enigma. What's not very well known about proteins, unless you have a chemistry background, is proteins are more than just a long, very long string of amino acids. Once that string of amino acids is built to the right length and the right sequence to build a protein, the next step to actually have it form into a protein is that long string of molecules has to fold into the proper three-dimensional shape. What's not very well known about proteins is proteins are three-dimensional molecules. They don't work until they're put together and then they fold. And they have to fold into the exact, precise, correct shape, three-dimensional shape, or they're not usable by the cell. They'll try to attach to where they're supposed to do their work, and if they're not the right shape, they just float off. They'll break apart, and those constituent parts will be reused by the cell to make another protein. So the selection and sequencing enigma of the building blocks. What this is you can have a bunch of letters, okay, but just a bunch of random letters, an A, B, a G, a W. Random letters don't make information, and they don't make a protein. So you have to select, in order to convey information, you have to select the letters that you need, but then you have to actually put them in the proper sequence. So if you have some A's, you have some B's, some C's, etc., that's not information. You have to sequence them into an information bearing sequence, like the cow jumped over the moon. You can have T's and H's and E's, etc., but unless you sequence them in the proper way, you don't convey information. So that's what we call the selection and sequencing enigma. You have to have information first in order to select the right letters to use, and then you have to know the sequence to build information like the cow that jumped over the moon from a random set of just letters. So these are just a few examples of the problems facing a naturalistic origin of life. We're going to sample a few and demonstrate the significance of intelligent design as a more viable alternative. So we're going to turn to the information or DNA enigma, which is also called the biological revolution. With the advent of Darwinism and later of neo-Darwinism, evolutionary biologists claim to explain that appearance in startling new terms. That's the appearance of design. Rather than explaining that appearances as the byproduct of design, they have contended that new forms of life arose from a purely undirected process. In On the Origin of the Species, published in 1859, Darwin argued that the adaptation of organisms to their environments, previously considered a compelling evidence of design, could be explained by natural selection acting on random variations, a process that only mimics the powers of a designing intelligence. Thus, in 1859, the appearance of design in living things has been understood by many biologists powerfully suggestive of an illusion, but an illusion nonetheless. As evolutionary biologist Francisco Ayala has argued, Darwinism consists of natural selection accounts for organisms and for their wondrous diversity uh, and of natural processes, the gradual accumulation of spontaneously arisen uh, variations or mutations sorted out by natural selection, a process that is creative uh, an alternative to a consciousness. Richard Dawkins is even more blunt, defining biology as the study of complicated things that give the appearance of having been designed for a purpose. Thus, whereas many physicists have recently discovered design by a super intellect as an explanation for the origin of the finely tuned features that make life possible in the universe, biologists have long resisted the design hypothesis. Ever since Darwin, they have assumed that they could, in the words of Ayala, explain design without a designer. Similarly, Francis Crick, who with James Watson in 1953 elucidated the structure of DNA, enjoined biologists constantly keep what they see was not designed, but rather evolved. It was, however, due in large measure to Crick and Watson's own discovery of the information bearing properties of DNA that the naturalistic understanding of life has begun to unravel. Scientists have increasingly, in some quarters uncomfortably, become aware that of at least one appearance of design in biology that has not been explained by naturalistic or any other purely naturalistic mechanism. The information present in every living cell. So the DNA enigma. When Watson and Crick discovered the structure of DNA, they made a shocking discovery. DNA information in the form of a 4-character digital code. Their model of DNA showed that strings of precisely sequenced chemical called nucleotide bases affixed along the interior of the DNA backbone could store information. Crick developed this idea further in 1958 with his now famous sequence hypothesis, according to which uh, the DNA of the nucleotide bases function just as letters in a written text or digital characters in a section of computer software. Just as letters or digital characters may convey information depending on their arrangement, so do certain sequences of chemical bases along the spine of the DNA molecule convey precise instructions for arranging amino acids from which proteins can be built. Crick knew that proteins perform most of the important life maintaining processes in the cell. For example, proteins function as enzymes catalyzing essential metabolic processes and reactions at rates much faster than would otherwise occur. They process genetic information and they form the structural parts of tiny molecular machines in cells. They suspected that specific sequences of bases on the DNA strand provided the assembly instructions for building the protein molecules in protein machines that the cell needs to survive. By the 1960s, a series of brilliant studies and experiments in molecular biology had confirmed Crick's hypothesis. By the late 1960s, molecular biologists knew where the information for building proteins was stored and even how that information was to build them, but they did not know where that information came from. I call this mystery the DNA enigma, mystery that is closely related to the question of how the first information originated. So the biological revolution is Watson and Crick discover the structure of DNA, and then they discover DNA is an information storage medium. Think of it as the hard drive of your body. It stores enormous amounts of information, the information you need to build, perform all the processes of life. So the double helix, that's we see here a drawing animation of what's called the double helix. It's like a ladder that's twisted in the interior of that ladder. Each of those rungs of the ladder is like a letters in an alphabet that store.
Imagine you discovered a stone with ancient words carved into it, and they told a really good knock-knock joke. What an archaeological find! But to your surprise, scientists concluded that it came about through purely natural processes. Just physics and chemistry acting on an exposed rock. What? You'd need very specific lines and shapes to form letters, and then those letters would need to be in a very specific arrangement to even get a bad knock-knock joke. Well, if you leave rocks out in the wind and rain for long enough, you're bound to get knock-knock jokes eventually. Nobody would buy that, but this is eerily similar to the typical explanations for the origin of life. Except life is no joke. Pun intended. And there are many more layers of complexity that would have to come together in order to kickstart life. To get life started, we need to produce the right building blocks in high concentrations and purity, combine them in the right way in spite of the nearly infinite set of unwanted molecules that natural processes are known to produce, collect them inside of a membrane to keep all the good stuff in and the bad stuff out, maintain homeostasis, harness energy from a constant and controlled source, and bring all of this together faster than all the individual parts would expire. Even if you could do all of this, you can't. You still wouldn't be anywhere close to starting life because life requires at least one more all-important and very difficult ingredient. Information. Paul Davies and Sarah Walker, professors of theoretical physics at Arizona State University, said, although it is notoriously hard to identify precisely what makes life so distinctive and remarkable, there is general agreement that its informational aspect is one key property and perhaps the key property. But what is information and where did it come from? Let's get into it. Where did information come from? That's easy. Nature can make information. Well, that's true, sort of. Nature can make certain kinds of information. For example, nature can make illusory information. When we see shapes in the clouds or a face in the waves, it's not really there, it's an illusion. And actually being created by our own imagination. That's not really a taco in the sky. Nature can generate repetitive or deterministic information. Think salt crystals or snowflakes or sedimentary layers. These structures are beautiful, but completely determined by physics. Those shapes have to be that way. Physics demands it. Nature can also produce indiscriminate or non-specific information. Think white noise, or a waterfall, or wind rushing through a canyon. If we came across those kinds of information, we would easily be able to explain it away as natural. We wouldn't need to look for any other explanation. On the other hand, we all know that nature cannot make a knock-knock joke or a speech like the Gettysburg Address. Those things are not illusory, they really exist. They're not merely repetitive patterns, there isn't any physical law that says this word must go after that word. And there were far more words that could have been included but weren't. And it does a thing, makes you laugh or informs you. This kind of information, specific arrangements of independent parts in a complex way, we intuitively know is beyond nature's capability to produce. In other words, if you found the Gettysburg Address in the forest, even if you knew nothing about how it got there or who wrote it, you would know there was an author. Here's the all-important question. Is DNA more similar to salt crystals and white noise, or to the Gettysburg Address? Scientists have known for years that DNA is an informational code that is used to manufacture proteins, the tiny machinery of life. But we keep learning how DNA contains far more information than just protein coding. DNA also regulates or controls what proteins are made and when. Now, this may sound easy or boring, but it's really important. Imagine you're a little E. coli bug. You're getting a little peckish and you want a snack. Your favorite food? Sugar, of course. Now, there's 2 different kinds of sugar you could eat, but every buggy knows that glucose is a way tastier sugar than lactose. Eating these 2 different kinds of sugars requires 2 different kinds of machinery. Sort of like eating soups requires bowls and spoons, and spaghetti requires plates and forks. If it's soup day, you don't want to waste energy making plates and forks. That's a dumb move. You're a smart little bug. Wasting energy makes you less fit, so you know to turn off fork production when you don't need it. This is regulation. Without this, all your precious energy would be spent making forks and spoons and spatulas and ice cube trays, and then you die. So, in the DNA, we have a library of blueprints for all the different tools we can build, but we can't build them all willy-nilly or we'll die. So also within DNA is information to build regulatory proteins as well as little docking stations for them to hook onto DNA. Normally, when glucose is available, no forks or plates are made. It's Soupville. But if there's only lactose, the lactose is sensed by a regulatory protein, causing it to detach from DNA, unblocking the code to build lactose machinery, plates and forks. The glucose sensor is the opposite. When it doesn't sense glucose, it binds to a different part of the DNA to advertise, hey, let's build some lactose machinery over here. So regulation of protein construction is another required layer of information in order to get life started. But wait, there's more. Those regulatory proteins also have regulatory regions of their own. Additional code in the DNA to control their production. DNA has code to regulate the regulatory process and can even have code to regulate the regulators. Multiple layers of coding complexity. When building proteins, of course there's code for which bits of the protein to assemble next, but there's also instructions on how quickly or slowly the next amino acid is mushed on there, which controls protein folding. If a newly formed protein isn't properly folded, it could be a complete waste, or worse yet, it could be toxic and kill you. And there's even more than that. Sometimes a string of DNA can carry one message when read forward, and a completely different one when it's read backwards. Like if Lincoln read the Gettysburg Address backwards, it was instructions on what he wanted for lunch. If you've ever tried to make a palindrome, it's like that, but way harder. The same string of DNA that codes for a protein can also contain embedded instructions on when to build it. Like if the first letter in every third word of Lincoln's speech also spelled out instructions on when and where he should deliver the speech. Plus, DNA isn't read one character at a time. It's read through a window, 3 characters at once. And if we shift that window over 1 or 2 spaces, we can sometimes get a completely different functional message. Imagine if the Gettysburg Address had another secret message in it. If you just moved the spaces over by 2, 407 years ago would become Fo usko rednies in resco. What if we just move them over by 1? Wait, what? There is a new message! Long story short is the best and you should totally subscribe. Wow! Thanks, Abraham Lincoln. Total bro, that guy thinking about us 160 years ago. Obviously, DNA is packed with information, and even that word information does it a disservice. What if someone says, we're talking about the origin of the first life here, not complex life? Life started out very simply. Information slowly accumulated over millions of years through chemical evolution, replication, random errors, and filtering by natural selection, just like this paper says. Well, there's 5 problems with that. Information can't accumulate because chemical evolution can't even get started. Molecules that store information don't naturally self-replicate. There are many well-understood properties of chemistry and physics that tell us a self-replicating biopolymer could not form naturally. And if it can't replicate, it can't accumulate. Any hypothetical self-replication process would need significant information to even begin. Number 2, Self-replication requires high accuracy in order to accumulate, like 99.9% accuracy. Lab experiments can't get anywhere near this. Number 3, things are constantly expiring. Just like you can't slowly store up a pool full of milk over a long period of time, having more time actually hurts because milk expires. You've got to do it in less time. This can't be emphasized enough. Time is not the friend to abiogenesis. Time is the enemy. And for the next 2 reasons, we need to know that the information that's required for life goes far, far deeper than a straightforward string of letters. There are multiple additional layers of informational complexity that are unique to the information in life, even in the simplest forms of life. Reason number 4 that the information can't accumulate over long periods of time. The information in life actually changes the same material that holds the information. Paul Davies and Sarah Walker said this: biological systems are distinctive because information manipulates the matter it is instantiated in. Okay, imagine if the words of the Gettysburg Address, when written as ink on paper, caused the paper to fold up and origami itself into a 3-dimensional shape that looked like Abraham Lincoln. Amazingly, the information of life is like that. For example, the information inside of some RNA molecules can detect if that RNA is needed, and if not, it will chop itself into pieces. DNA contains self-referential information. It can sense it has, say, 2 of this gene, and then decide, let's have 100. It can modify the number of genes stored within itself. This is wild. And number 5, this information can't accumulate because the information of life is not just centralized in DNA. It sounds crazy, but it's also kind of obvious. DNA can't do anything on its own. It can't even replicate on its own. It'll just sit there. The information needed for life is scattered across various parts of the cell that are completely dependent on each other to survive. This creates so many chicken and egg paradoxes, it's not even funny. DNA can't do anything without RNA and proteins. Proteins can't exist without DNA and RNA. RNA comes from DNA via the help of proteins. You need them all at once. Paul Davies and Sarah Walker again. The manner in which information flows through and between cells and subcellular structures is quite unlike anything else observed in nature. For example, DNA doesn't contain the information to construct a cell membrane, only to add to it. The information that started the first cell membrane has instead been passed on from generation to generation, membrane to membrane, circumventing DNA. The cooperation between sources of information in life is inherited, passed down from generation to generation. All of the different connections needed between these things, they're not held within DNA itself. It just exists already and is passed down. Not to mention there's many other codes of life, not just DNA. More on that in a later video. So is the information in DNA in the nature could have done it camp or the nah camp? Unguided natural processes can't produce jokes or speeches. They are beyond nature's capability. It isn't even a question. If you see something like the Gettysburg Address, you immediately know someone produced it. Even if we never saw the author, even if we know nothing about them, an author must exist. The information needed for even the very simplest life is not merely like a knock-knock joke or a speech. It far surpasses it. In many areas of science, investigators have to decide if natural processes or an intelligent mind is the best explanation for their observations. Archaeologists look at buried artifacts to determine if they were made by natural processes or a creative mind. Are these arrowheads or just rocks? Coroners investigate deaths to determine if they were caused by a natural process, like old age, or by the will of a human mind, homicide. SETI looks for signs of intelligence in outer space. These scientists must remain unbiased when deciding between natural processes and intelligent causes. But somehow, when it comes to the origin of life, scientists can only consider purely natural explanations. Thinking otherwise is strictly prohibited. Wouldn't it be better if we were allowed to follow the evidence in science wherever it leads, rather than being forced to follow the presuppositions and preferences of the scientists? Thanks for watching.
So we see information is critical to the origin of life. You can't separate information and the formation of life. So the question is, from what we've been presented, is where did the information come from? Nature can create some information, like crystals and snowflakes, but the information that is in those crystals and snowflakes looks more like the following, like AAA, BBB, CCC, or ABCD, ABCD. It's repeating non-specified information is what it would be called in the literature. And this would be very different from something like the cow jumped over the moon, which is specified information that conveys meaning. So also the information in DNA contains gene regulatory networks. Those are the cell's on-off switches, which means there's processes that goes on in the cell, such as building a certain type of protein. Well, when there's enough of that protein, and the cell needs to stop making that protein and make something else, there are information feedback mechanisms within the cell that tell the cell to do that. Stop producing one thing and then go on and produce another. So the cell also has layers of information, backwards, forwards, different formats in the DNA, communicate and store even more information. So we'll just quickly review our video for the 5 problems of chemical evolution. It can't get started in the first place because for chemical evolution to begin, it needs replication already at the beginning. You have to have self-replicating molecules, and replication requires information. So the information has to be there in the first place, and where did it come from? So information is required at the beginning. Self-replication requires high accuracy. You can't go through a series of mistakes or trials and errors because of the precision required to replicate the detailed information in the cell. Things are constantly expiring. Time is the enemy of chemical evolution. As I said previously, if those chemicals are in a water-based environment, they'll fall apart because of hydrolysis. If they're exposed to sunlight, they'll decay. If they're exposed to oxygen, they'll decay. So organic molecules are delicate and they don't stay around for very long. Organic molecules like those in the cell are delicate and decompose very easily. The information in life changes the material that holds the information. So once DNA is programmed or stores some information, the physical molecules themselves are rearranged by the information. So in the information of life is not just centralized in DNA. There's different places within the cell that have the information. So you have to have all that information there, all complete from the beginning. So we'll watch a video here, another video of some molecular machines within the cell as a demonstration of the complexity of the cell, but also the incredible amounts of information that are needed right from the beginning.
These are tiny molecular machines, and they are doing this inside your body right now. To understand why, we have to zoom out. Every day in an adult human body, 50 to 70 billion of your cells die. Either they're stressed or damaged or just old. But this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells, right now, billions of your cells are dividing, essentially creating new cells. And that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines. So let's take a closer look. DNA is a good place to start, the double helix molecule we always talk about. This is a scientifically accurate depiction of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has a sugar phosphate backbone connected to the sequence of nucleic acid base pairs, known by the letters A, T, G, and C. Now the strands run in opposite directions, which is important when you go to copy DNA. Copying DNA is one of the first steps in cell division. Here, the two strands of DNA are being unwound and separated by the tiny blue molecular machine called helicase. Helicase literally spins as fast as a jet engine. The strand of DNA on the right has its complementary strand assembled continuously, but the other strand is more complicated because it runs in the opposite direction. So it must be looped out with its complementary strand assembled in reverse, section by section. At the end of this process, you have two identical DNA molecules, each one a few centimeters long, but just a couple nanometers wide. So to prevent the DNA from becoming a tangled mess, it is wrapped around proteins called histones, forming a nucleosome. These nucleosomes are bundled together into a fiber known as chromatin, which is further looped and coiled to form a chromosome, one of the largest molecular structures in your body. You can actually see chromosomes under a microscope in dividing cells. Only then do they take on their characteristic shape. Otherwise, the DNA is more strewn inside the nucleus. The process of dividing a cell takes around an hour in mammals, so this footage is from a time-lapse. You can see how the chromosomes line up on the equator of the cell. Now, when everything is right, they are pulled apart into the two new daughter cells, each one containing an identical copy of DNA. Now, as simple as this looks, the process is incredibly complicated and requires even more fascinating molecular machines to accomplish it. So let's look at a single chromosome. One chromosome consists of two sausage-shaped chromatids, containing the identical copies of DNA made earlier. Each chromatid is attached to microtubule fibers, which guide and help align them in the correct position. The microtubules are connected to the chromatid at the kinetochore, here colored red. The kinetochore consists of hundreds of different proteins working together to achieve multiple objectives. In fact, it's one of the most sophisticated molecular mechanisms inside your body. The kinetochore is central to the successful separation of the chromatids. It creates a dynamic connection between the chromosome and the microtubules. For a reason no one's yet been able to figure out, the microtubules are constantly being built at one end and deconstructed at the other. While the chromosome is still getting ready, the kinetochore sends out a chemical stop signal to the rest of the cell, shown here by the red molecules, basically saying this chromosome is not yet ready to divide. The kinetochore also mechanically senses tension. When the tension is just right and the position and attachment are correct, all the proteins get ready, shown here by turning green. At this point, the stop signal broadcasting system is not switched off. Instead, it is literally carried away from the kinetochore down the microtubules by a dynein motor. That's the walking guy. This is really what it looks like. It has long legs so it can avoid obstacles and step over the kinesins, molecular motors that walk in the opposite direction. Personally, I'm astounded by these tiny molecular machines, how they're able to routinely and faithfully execute their functions billions of times over inside your body at this exact instant. I'm also amazed by the scientists who were able to work out how this happens in such detail that we could create realistic depictions of them like you saw in the animations in this video. But perhaps the most amazing thing is just how much is left to be discovered, like figuring out how exactly the chromatids are pulled to opposite ends of the cell. There is still so much that we don't quite know. You know, what I find exciting is that in science fiction, for decades, we've been writing about tiny nanobots that will be injected into our bloodstreams that can heal us. But what this suggests, the existence of these tiny molecular machines inside us, it suggests that there isn't a physical limit that would prevent that. And so I think it's pretty likely that in future, we will be able to develop our own tiny molecular machines that will be able to repair our bodies better than they can repair themselves.
So what you're looking at on the right is a protein called kinesin. Those red kind of bulbs at the bottom are actually feet, and the round object that it's attached to is representation of a molecule that kinesin is transporting from one part of the cell to the other. And what it's walking on is called a microtubule. So, how did these machines evolve individually? They couldn't be, it can't be done by trial and error, because for the cell to be alive, all these nanomachines have to be functioning. All these machines need to be functioning and functioning properly for a cell to be alive. How did life begin if all these machines were evolving from non-functional states? How does the kinesin machine know what molecules to latch onto and where to bring the molecules it's transporting? It needs information, feedback, and detection systems. Where are these found outside the body? Well, they're found outside the body in man-made machines called factories. If you produce products, you have information feedback systems. So we see where we have objects that are or factories that are designed or created by intelligent designers have these information feedback mechanisms. And how does kinesin know when to release the molecules it's carrying? It has to go to one part of the cell, but does it know what part of the cell to go to and when to release it? Where did the information come from to build these machines? Now you can begin to see the information enigma. Where did the information come from? So, as Bernd Olaf Kuppers stated, the problem of the origin of life is clearly basically equivalent to the problem of the origin of biological information. Do we know of any entity that has the causal powers to create large amounts of specified information? We do. As information scientist Henry Quasler recognized, the creation of new information is habitually associated with conscious activity. Experience affirms that functionally specified information routinely arises from the activity of intelligent agents. A computer user who traces information back to its source invariably comes to a mind, that of a software engineer or a programmer. Similarly, the information in a book or newspaper columns can be traced back and derives from a writer, from a mental rather than strictly material cause. The actions of a conscious and intelligent agent clearly represent a known and presently acting and adequate cause for the origin of information. Uniform and repeated experience affirms that intelligent agents produce information-rich systems, whether software programs, ancient inscriptions, or Shakespearean sonnets. Minds are clearly capable of generating functionally specified information. Further, the functionally specified information in the cell also points to intelligent design as the best explanation for the origin of biological information. Why? Experience shows that large amounts of such information, especially codes and languages, invariably originate from an intelligent source, from a mind or a personal agent. In other words, intelligent activity is the only known cause of the original origin of functionally specified information, or at least starting from a non-living source, that is, from purely physical or chemical antecedents. Since intelligence is the only known cause of specified information in such a context, the result of functionally specified information sequence in even the simplest biological systems points definitely to the past existence of an activity of a creating intelligence. So, the information enigma shows us evidence for an intelligent designer, clearly. So next week, we're going to go over, the question is the Bible reliable? And we're going to delve into the art and science of history, how history is done, and how we can apply it to the Bible. So Once again, to recap, the sources for today's materials are The Mystery of Life's Origin, Signature in the Cell, and Unlocking the Mystery of Life. We'll see you next week.